I'd like now, on behalf of everyone here and people watching the recording on YouTube, I'd like to introduce John Roberts and to thank John for offering to deliver this presentation, which is very topical. So welcome, John.
No problems. Yeah, and thanks for having us along. I've been working with CSIRO for around 15 years now, looking at bee pests and pathogens. A lot of my work has been around molecular techniques, particularly around surveillance. And so today I'm wanting just to share a bit of the work I've been doing looking at eDNA and how we could be using that for biosecurity. eDNA stands for environmental DNA. And it's really just the traces of genetic material that are left in the environment by the different organisms that are living there. And we have a range of different methods that we can collect eDNA from aquatic and terrestrial environments. And then we use genetic methods to identify the species in those eDNA samples. And so for me, I kind of think this is like environmental forensics a bit. What this figure is trying to capture, I guess, is the different applications that we can be using eDNA for. Typically, we're thinking around questions of biodiversity, but more and more we're looking at ways that we can be using it for biosecurity applications and detecting invasive species. Beehives are a really good source of eDNA. My research has been focused on using Hive eDNA for some of these biosecurity applications. And so for me, what makes beehives so good is that every beehive has thousands of foraging bees that are going out potentially for several kilometers out from their hive, and they're interacting with that surrounding environment as they're going about collecting nectar and pollen from flowering plants. Some of these plants could be weeds of interest. They could perhaps be infected by plant pathogens. And the foraging bees are going to bring traces of those plants and those microbes back to the hive, and they're bringing them back in the form of eDNA. And so that gives us the opportunity to collect some hive samples and collect that eDNA. So common sample types that we use are often a bee sample, but we can also be looking at things like honey and pollen, and something that I'm talking a bit more about today is even the use of swab samples. So very much the COVID type swab, but using that for swabbing the hive surfaces for eDNA collection. Once we have our eDNA sample, we then use our genetic methods for identifying what the pests and pathogens that are in those samples. We have methods like high throughput sequencing, which are used for more broader species detection, but we can also use more targeted PCR approaches, which are designed to identify specific species. So at CSIRO, we've been using both of these methods to develop Hive eDNA surveillance, particularly for bee pests and pathogens like Varroa mites, and that's what I'm mainly talking about today. But we've also been looking at Hive eDNA as a tool for detecting things like plant pathogens and weeds. And so I'll spend the last part of my talk just touching on some of that work as well. Surveillance for parasitic bee mites. It's a key part of bee biosecurity, and typically we're thinking around early detection for our exotic mites, but we've now joined the rest of the world, and we also need to be using these methods for monitoring our mite levels and for management. And we have a range of visual type methods where we're trying to collect mites and observe them. And these are pretty good methods, but we do know that they can be more labor intensive, and they're often can be limited in their sensitivity. I'm sure we're pretty familiar with some of these methods. Things like drone uncapping is certainly quite easy, but the sensitivity is going to be much lower than some of our other methods like an alcohol wash or sticky mats, which do take more effort. Our other picture is a dissected bee, which we would use for looking for tracheal mites, which is an internal parasite, and that's definitely more labor intensive. So when we're looking for mites, while we do have a number of methods available, I think there's a real opportunity to be using eDNA methods in the mix as a complementary tool and try and have something that's potentially more efficient and a more sensitive approach. With some collaborators at the University of Canberra and also in the Ministry of Primary Industries in New Zealand, We've designed and established a range of genetic tests, so PCR tests that are species specific for our different bee mites. They're designed to be effective for detection in eDNA samples. And so this first step, which is based in the lab, it's really about making sure that these genetic tests are both species specific so that they have a low chance of false positives, but we also want them to be highly sensitive so that we reduce our chance of getting false negatives. We've done this lab work. We've been happy with how they're performing in the lab. What we would then want to see is how they're working with real life samples, with our hive samples. And so we have had a project where we've been able to explore this more, looking at Varroa destructor. This was a project funded by DAF. When we first started this experiment, we were still in the middle of our eradication response for Varroa in Australia. So it was not easy to get access to areas from those infected zones. So we were lucky to be able to work with some colleagues at MPI and do our experiments across over in Wellington in New Zealand. Varroa is pretty well established there. They've had Varroa mites since 2000. So with the help of MPI staff and local beekeepers in Wellington, we're able to test out some of our eDNA detection methods that we're using both honey samples and surface swabs from the hive entrances and from brood frames. And we're comparing that to a conventional alcohol wash for detecting mites. With the honey sample, we're collecting a sample of 50 mils from a single frame within the hive. And for the swab samples, we're collecting duplicate swabs, 2 swabs from the hive entrance and 2 from the brood frames. For one of the experiments, we had 42 managed beehives that had a range of mite levels. They had an average of about 7 mites per 300 bees in the alcohol wash. So that's about a 2% infestation level. And what we were really pleased to see is that the eDNA detection methods were working quite well. All 3 of them were detecting Varroa eDNA, and we're seeing that our qPCR amplification was increasing as the mite levels were increasing. So what this means is that we're seeing more eDNA where there were more mites. We also want to be able to compare the sensitivity of the different methods. And so for this, we assume that all of the hives had mites so that we could compare across to the alcohol wash. And what we were finding is that our honey sample had the highest sensitivity, 91%. This was because we were getting eDNA detections in our honey samples from hives where we weren't seeing anything in the alcohol washes. The brood frame swabs was showing comparable sensitivity to the alcohol wash, which is good. And our hive entrance swab samples were showing lower sensitivity. We're getting false negatives in those samples. And when we looked in more detail at the data for those samples, we could see that these false negatives were generally from hives that had very low mite levels. From this experiment, both the honey samples and the brood frames were looking like a good surveillance approach, but from a practical perspective, they still required a bit more work for collecting those samples. So we really did like the hive entrance swabs in that they were non-invasive. We were able to use that method without opening the hive. Another experiment we did over in New Zealand was where we were specifically wanting to look at eDNA detection during that early mite invasion. And so for this, we brought 10 nucleus colonies across from the Chatham Islands, which is to the west of New Zealand and is still Varroa free. And we brought them across to the Wellington Club apiary and monitored them as they first became invaded with mites. So for this experiment, we only collected eDNA swab samples from the entrance and brood frames for the first 8 days. And then from day 8 onwards, we were also doing the alcohol washes. And this was designed to try and limit the impact on these small colonies. In this experiment, what we found was that all hives had become infected within the 2 months of the experiment, which was good, also a little bit scary. What was also really nice to see is that we were detecting Varroa eDNA either earlier or at the same time as the alcohol wash method in 8 of those 10 hives. So this was giving us some confidence that we can really detect those early mite levels. But again, we're still seeing some false negatives. So 2 of those hives, 2A and 4B there, we had mites detecting the alcohol wash, but we didn't have any eDNA detection during the experiment. So we had a lot of fun and generating our New Zealand data, but we were keen to get some Australian data as well. And so last year in April and May, we had a chance to test out our eDNA approaches with some New South Wales hives. For this, we had 2 apiary sites that had 70 hives each. And in this experiment, we only used the hive entrance swab method, but we did this at 3 time points. What was nice to see with this experiment is that our results were fairly similar to the New Zealand experiment, where we were seeing an increase in the eDNA amplification when there was an increase in mite numbers, but the eDNA sensitivity was a bit mixed. Overall, it was generally comparable to the alcohol wash, but what we can see in that first time point is that the eDNA sensitivity was really quite low. And so at this point, our mite levels were around 1%, so pretty low. But by the time we got to our second and third time point, the eDNA sensitivity was looking quite good. And so our mite levels were above 2% at that stage. We really like the efficiency of the hive entrance swabs. They're really a simple approach and being non-invasive, really a method that we can see as being a scalable approach for surveillance. But we still really want to get a detection at those low mite levels improved. We haven't yet been able to do some more experiments on that, but we think that we probably can address that with some pretty simple increases to sample replication. By collecting more swabs more often, it's going to give us a greater chance of collecting those low concentrations of Varroa eDNA when the mites are at really low levels. The honey samples were also looking really good for collecting eDNA for Varroa, but the practicalities of doing that at the individual hive level were not there. So we think there's a better approach for using honey and using that more for broad scale surveillance, where we're using already extracted honey and using that as an early surveillance tool. That's really something that I'd like to see these eDNA methods being adopted for, particularly in our non-Varroa areas, such as WA and Tasmania, and using, I think, both the honey and swabbing methods can be a really nice complement to our other surveillance approaches. We have been asked and thought about whether we can be using these kinds of methods for our routine mite monitoring to inform our management. But I think at the moment we need a little bit more optimizing around that. I think the cost and the turnaround time for results is probably still a bit limiting if we want to use it to inform our management decisions. And then something else that I really want to see this expanding to is using it for broader surveillance of our bee pests and pathogens. I think there's lots of opportunity to be using it for things other than Varroa. We have already started looking at using hive entrance swabs to detect other mites, so Varroa jacobsoni and Tropicalaps mites, and seeing some really promising results with that as well. Using these tools more broadly in our biosecurity system, I think, is only going to improve our sensitivity and efficiency. I just want to finish with some other experiments we've been doing that's exploring the use of hive eDNA more broadly for biosecurity. And one of those experiments was doing a comparison of some common hive sample types for the broader eDNA detection. So with this experiment, rather than using the species-specific detection, our PCR testing, we're using high-throughput sequencing approaches that capture the broader range of species in those samples. What we're finding with this experiment is that our hive swab methods were again performing really well, which was a little bit surprising to us. We were pleased with what we're seeing with the Varroa experiments, but we were a little surprised at how well they're performing in detecting all the other types of taxa that we're interested in. So in this experiment, we're looking for bacteria, fungi, other invertebrates, and plant species. The graphs on the left there are looking at the total number of taxa, and our graph on the right is looking at the replication. And so what this is showing is that with only 2 replicate swabs, we were collecting the majority of the eDNA diversity in these samples. So this is looking quite promising. We also were interested in how we could be using these different sample types for detecting viruses, and we're seeing a similar result where the hive swabs and also the honey were performing really well. Overall, this was helping to demonstrate how we can be using these eDNA methods, particularly the swab samples, as a way for broader biosecurity surveillance using hives. Lastly, I just wanted to touch on another experiment where we're putting our swab method into action and investigating how we can be using Hive eDNA from urban hive networks to support weed management. For this experiment, we were collaborating with the ACT beekeepers to collect hive entrance swabs from their hives in October and November. We were comparing the plant eDNA detection results with the ACT government's weed management records. So we're still analysing this data, but some of the initial findings is that we're finding that the Hive eDNA data was detecting a large number of weed species. We're finding several weeds of significance. Another result is that we're able to get some understanding of how well that correlation of the number of hives sampled and the number of weeds. And so the more hives we're sampling, we're getting a greater number of weeds detected. Again, just highlighting that not every hive at a location is going to be foraging on the same things. Graph on the right here is a comparison of our eDNA detections against the management records over the last 10 years. The part of the graph on the left would be our weed species that are either not managed at all or we only have a few management records for, and then we have weed species on the right-hand side there that are more highly managed. We're still trying to unpack this data a little bit. Species that were fairly common in our eDNA detection, things like clovers, as you might expect, but they're not really being managed. Whereas there were other species that were in a sample set that were commonly being managed by the ACT government, but we weren't picking them up in our eDNA data. And these were largely grass species such as serrated tussock. Again, this is not too surprising from our hive samples. And then we have some of these other samples that are also really important weed species, and we have a lot of management records for, and we were detecting them frequently in our eDNA data. And so what we're still analyzing this data for is trying to understand how we can inform those management decisions. Is the eDNA data suggesting that we have areas that need more management, or perhaps it can be informing that management's been effective in certain areas? Just a quick summary, we have some robust eDNA methods for detecting Varroa mites, particularly from honey and hive swabs. I think we can be using these methods already to support our surveillance approaches. There's lots of opportunities to be integrating Hive eDNA approaches into our national bee pest surveillance program. We've got our sentinel beehives at ports. And just something that I was wanting to highlight with that last experiment is that we also see lots of opportunities for being able to scale up our surveillance in Australia by using these urban hive networks and working more with our amateur beekeepers to have broader biosecurity surveillance.
Thanks very much, John, for a most interesting, relevant and thought-provoking presentation.